Go
Why
ahead.
do you label the UTPs instead of the NTPs, or why do you use the UTPs to label the probes?
I think someone asked me that earlier at one point.
Just I've
I never figured out the answer, but my guess is biochemically, it might be easier to purify the UTPs. Maybe they're more stable. Maybe easier biochemically to label.
I see.
I think it's my guess. Yeah, I should look into more, but I don't have a real answer for that. But typically it's the UTP. Sometimes I think people use DCTP as well as another option. So keep this in mind as we're going through the different assays. We've covered karyotype and fish. So I listed some of the downsides, right, of fish. The one downside of fish versus karyotyping is that you don't get that genome-wide coverage that you would get by karyotyping. What you do sacrifice, you do gain in terms of resolution by fish, and also in terms of specificity if you were to able to design your assay for the rearrangement or the copy number that you're looking for. Whereas karyotype relies on a very large region to be changed. So next I'm going to talk about array CGH, which kind of gets you back to the mode of karyotyping with some caveats. So array, comparative, genomic, hybridization. So this is array, comparative, genomic, hybridization. So this is John's claim to fame, his 2003 paper, which was one of two papers that came out. I think one in Nature Genetics, which is John's paper. The other one was Science. Forget the name of the group. But these two papers were the first proof that human genomes differ very drastically from one another. Two individuals, if you were to compare them, there are large chunks of DNA that are gained or lost, and they are quite different between those two. And it explains more variation than if you summed up all of the SNPs. So typically I think you have about 3 million SNPs between two different individuals. But array CGH would identify copy number changes that are way more than that in terms of copy number change. So the technology to detect them is array, comparative, genomic hybridization. So let's start first with array. An array is essentially just a matrix of different entities that can allow you to do very highly complex and high throughput, massively parallel sort of processes. So you can think of arrays as tissue microarrays. There you have many different cores on one array that you section through, and you're trying to do immunohistochemistry or FISH or some other sort of in situ assay in terms of analyzing across multiple different specimens at the same time. You can think of protein microarrays as a way to test maybe antibodies, or the vice versa. If you have antibodies coding arrays, you can analyze many different protein analytes at the same time by using a protein microarray. Technically, there are no RNA arrays, but there are DNA arrays that can look for RNA expression. If you take RNA, convert it to cDNA, and have an oligo chip that recognizes different genes and different exons that you're trying to look for, you could assess for gene expression in terms of copy number of alleles that are being expressed by gene expression. So what we're going to talk about today are DNA microarrays are used for detecting dosage of DNA. How many copies of DNA do you have in a patient relative to a control? So the array is essentially just a glass slide. You can imagine there's a grid on there. And I'm going to blow up one of these spots right here, which maybe is this square right here. Within the square, you can imagine there's a circle which defines where the oligos are covalently bound to the glass slide. So here, the oligos are kind of sticking up like this. They're all monoclonal, or they're of the same species. And typically, depending on which array you're looking for, so there are arrays that are used for SNP genotyping, which these oligos tend to be much shorter. They're about 25 mers. And famous ones are the Affymetrix ones, for example. Illumina also makes SNP genotyping arrays as well. But for those that do copy number, they typically are a little bit longer, 50 to 60 mers. So this flavor is from Affymetrix. This flavor is from Agilent, which is the technology that we use. Arguably, you can actually use 25 mers to also do copy number detection, but it's a little bit noisier because you have shorter stretch of DNA, technically less specificity than just 50 or 60 mer. If you get up to 50, 60 mers, it's very hard to distinguish a SNP. If you have one base change out of 50 or 60, it's harder to do. So that's why for SNP detection, people typically do 25 mers. But today we're going to focus on this section right here. And let's just say these are 50 or 60 mers that perform very well. So what will happen is Agilent will go through each one of these spots here, and using a process called photolithography, they can print, kind of like an inkjet printer. They can print very specifically on this one spot the 50 mer that belongs to this certain coordinate in the genome. So it's a very specific probe for a certain coordinate in the genome. And there will be hundreds of thousands to millions of these oligos all sticking up. And it will be just one sense. It will be either on the positive strand or the negative strand, depending on how you design that probe. And you take this process and multiply it by 180,000 times, or some chips can have up to 1 million features, for example. So depending on how deep of a resolution you need for your detection, you can go very high or minimally high or medium or whatever. What we use is a chip that allows you to do 4 assays at the same time or on 4 different patients. So these are kind of like quadrants or whatever, 1, 2, 3, 4, and each one has 480,000. Each one has 180,000 probes on them, or 180,000 features per quadrant, and you can do each individual patient in each of these sort of quadrants right here. So the process is that you have this array. It has tons of these oligos, each one representing a unique feature in the genome that you're trying to detect. And this is essentially one of the substrates that's needed for array comparative genomic hybridization. Cost-wise, my feeling right now is probably each one of these will cost about $250. So it's one chip that's probably $1,000 or $1,100. Each one is about $200, $250, or $275 or something like that. So it's not a cheap assay. It's one of the more expensive assays that we run in our lab. And arguably it's the most complex assay that we run because essentially you could design this to cover the entire genome in intervals. You're not really covering every base in the genome. You're covering maybe every 10,000 bases. There's a probe that hangs around to do copy number. So the resolution is not going to be down to hundreds of bases or 1,000 bases. It's really going to be dependent on the minimum call that we make and how many probes that cover and how well spaced they are. So that's the array component. So I'm just going to draw that one spot here with the oligos. So let's see where the comparative comes from. The comparative is you're going to take 2 DNA samples. You can take the patient DNA and you're going to take control DNA. So the last lecture is kind of nice because we kind of can segue into array pretty easily. So remember the random prime labeling process that I talked about to make back clones and use back clones to make probes for fishing or fish, the fish assay. You can use that exact same process and take patient DNA and control DNA and label them differentially. So in one sense, you use a DUTP that's labeled with red. So you label the patient in red and you label the control in green. So it's the same process, random hexamers, random prime labeling, and it just uses the patient DNA as the template or the control DNA as the template. And it makes a new daughter strand, nascent DNA strand that's either labeled red or labeled green, depending if it's patient or control. You do this in 2 separate tubes. So this will be going on in this one tube, second tube. So up front of this, some folks like to do some shearing because shearing helps the DNA denature during this process. So you might use enzymes, for example. Agilent recommends using 2 enzymes, ALA-U1 and RSA1. And I'll get into a bit why that's needed later. You can also do mechanical shearing, so if you did sonication, sort of a non-specific way of shearing the DNA, you could cut it down to maybe 200 base pairs, 300 base pairs, maybe 150 will work as well. And then after that, allow those to melt, serve as template for the random prime labeling to label the patient DNA or the control DNA, red or green respectively. So after labeling, you're going to do some cleaning process, and you're going to mix them together. You mix the two samples together. Hopefully, you have already done an upfront step to normalize the two samples. Typically, we like to do qubit analysis so that you're quantitating equal amounts of double-strand DNA between the two samples. Put in 1 microgram of each sample during the labeling, and then after labeling, after cleaning up, you're going to combine the two samples together and mix them. And what you allow them to do is allow them to hybridize. So here's the hybridization step. You allow them to hybridize to the target DNA on the spot on the chip. One of those 180,000 spots that are on there. So during this process, what is happening is that the mixing of the two samples allows a competitive environment for you to do comparison between the patient versus control. So we have an array. We're comparing two pieces of DNA, one from patient, one from control, and we're allowing them to hybridize to the chip right here. And so when you do that, let's say you start out with, just an example, 1,000 copies of the red and 1,000 copies of the green for some particular region of genome represented by this one oligo right here. If you have equal molar representation of the two, in the end you're going to get equal binding, equal chances of binding between the red and the green. And when you go to do the analysis, it's going to show up as an intensity that is equal between the red and the green after you normalize, which means that you have a copy number neutral state. So the dosage in the patient is no different from that in the control. If you have, say, more of the patient, I'm going to draw in red now, let's say you have 4,000 copies of something, versus in the green here you have only 2,500. Obviously, this has more chance, more molecules to participate in hybridization, so therefore you can get more red signals binding than green. And then when you go to do the analysis, this will show as higher red intensity, lower green intensity, and there's a ratio between the two that will kind of guesstimate how much dosage you have in the patient relative to control. So all this relative hybridization and binding is the comparative step in array CGH. Genomic is what I've already mentioned, in that you could design this array so that it covers the entire genome in intervals of every 10,000 bases or so. So certain chips are designed that way to be sort of naive to disease, but others, like the ones that we're using, is developed by a consortium called the International Standards for Cytogenomics Analysis, or ISCA. So they have a target disease gene list of about 500 or so. 500 disease regions in the genome that they heavily design probes for and dedicated to those regions. Probably guaranteeing, I forget, 20 or 50, at least 20 or 50 probes in those regions to get better resolution for those disease sites. And then the rest of the probes are just the backbone probes, which are scattered about every 10,000 bases between each one. So obviously the disease regions will be much higher resolution because it's more packed and more dense. And the rest of the backbone will be a little bit more sparse because there's more spacing between the different probes. So that's where the genomic comes in. So essentially you do this, allow to hybridize, we're doing now 48-hour hybridization at, I believe, 65 degrees is the temperature that we're doing. And during this process, what happens kind of technically is you have those 4 quadrants. On the chip, and what you do is, this is the chip, you're going to put a cover slip or another slide on top to cover slip it, and this cover slip has sort of these gaskets, so it's the same rectangular shape here, 4 sets of them, and then when you press them together with this sort of contraption that essentially kind of squeezes the two together in sort of a pressure type situation. What you have is 4 individual hybridizations that occur. So the liquid is trapped in here. It's roughly 100 microliters or so, where you mix the two samples, some hybridization solution, some blocking solution as well. But you put this slide into this rotisserie that operates at 65 degrees temperature and is rotating constantly during this 48-hour incubation or hybridization. So thereafter, you're going to remove things, wash the non-specific things out, and then you kind of cover, it's not really cover slipping, but you put it into this other protective, ozone protective barrier contraption, and then you're ready to scan it. So a lot of this sounds very similar to fish, right? So all of the probing, the washing, et cetera, is essentially the same as fish, but it's technically fish in reverse. So whereas in the in situ assay, we have the patient DNA immobilized here, waiting to be probed by some colored probe. So let's say there's a red probe getting showered down here. In array CGH, it's the opposite. It's the patient DNA that becomes sort of the probe by being labeled, but what is being immobilized is on the probe itself that's sitting on the chip. So you can basically flip it in reverse, and that's what array CGH is. And so in the end, array CGH can allow you to detect things that are actually as small as fish, sometimes even a little bit smaller, but it's all contingent on how you design the spacing of the probes. So any questions so far about the technical protocol? So you do the hybridization, you wash, and then you scan, and we'll talk about the resolution and what that depends on. So what you do is you go through each spot and you scan it. And you look for the red signal versus the green signal. And you're going to express this as a log-based ratio of 2 between the red and the green. So in the scenario where I mentioned earlier, perhaps you have, let's just say at the cellular level, the red patient or the patient DNA shows that they have 4 copies of something. And the green patient, or the green control, sorry, has 2 copies of that same thing. So when you do the math, this is going to be log base 2 ratio of 4 to 2. This is going to be the same thing as log base 2 ratio of 2, right? And that equals 1. So if you have something where it's log base 2 ratio of 2 to 2, a copy neutral state, this is log base 2 ratio of 1. The answer is 0, right? So what we do is we go through each spot on the array, and here's the array again. You just go through each spot, and you just count how many red signals do you have, how many green signals do I have. The intensities between the two are going to be a little bit different. Green may be a little bit brighter than the red, for example. So the computer will know to normalize that based on looking at all the probes. It's going to normalize it back down between the red and the green and make it a little bit more equal and more like each other in terms of intensity. And then you do this ratio business. And what you do is you take adjacent probes. So here on the slide or the array, you have those 180,000 probes. The way it's designed is that this probe right here is not going to be right next to that probe. You don't want to design that way. You want to spread it kind of randomly across the chip. And the main reason is if you have some bubble or something that happens right here, or some lint or dust that contributes to the signal, you don't want all those neighboring probes to be right next to each other because it's going to give you a false positive call. So instead, you're going to spread them out. So maybe this is the coordinate that's right next to this one, and you kind of distribute it out. So you do across all of that, and you rearrange them back in order in terms of genomic position, and you express it as this ratio here. So what I'm going to draw is a log base 2 ratio plot, like that. So for most cases, let's just say we go through, right, and we have 6 or 7 probes that all are not showing any difference between red and green. So what does not any difference mean? We usually have a threshold. It's at 0.25 or minus 0.25. So I forget what 2 to the 0.25 is. 2 to the 0.25 is 1/8. Is that right? So if there is a little bit more of something, 1/8, am I getting this right? That's the negative, sorry. Apologize for that. It's not 2 to the 0.25, but what it is that there's a certain level, let's say you have 1.2 of patient relative to 1 of the control. So it's not exactly a 1 copy gain, it's just maybe under a 1 copy gain. It will give you some fraction like this. It doesn't really make sense, right? When you talk about germline conditions, for example. Unless you have a situation where patient shows 5 copies of something and control shows 4 copies of something. Then you might have that kind of weird ratio. But anyway, we allow for some breathing room between this 0.25 number and minus 0.25 number to call something normal or copy neutral. So we'll color these black for right now. Let's say you run into the situation where now you really have a legit copy number gain for this one probe. So this one is showing more red, so I'm going to now draw a dot around the 1 level. So now you have 1 dot that's legitimately gained in the patient relative to the control. Now let's just say the next one is back down to normal. So typically when I see this, it may be real, but I'm not going to trust it because it's only 1 data point out of 180,000 or so that's saying that there is a copy gain here. But I'm not going to believe just 1 data point. So I'm going to let that go as noise, even though it could be legit and real. The assay the way it's designed is not to allow you, it will allow you to detect that, but it's something you should not believe analytically. However, let's say I go through and then there's another set of probes. There's 5 of them that all show loss to some degree like that. 5. And then things get back to normal. So now what I have is 5 data points that exist at various places on the chip that's all screaming to me and saying, yep, I have a loss here. And the loss is a -1. So 2 to the -1 equals 1 over 2, right? So this means that in the patient I have 1 copy in the patient and I have 2 copies in the control. So there's a 1 copy loss. I have now 5 data points that say, yep, there's a loss there. This, if you were to calculate the p-value, the chance of this happening, depending on the noise and how things are distributed on your array, is highly significant. So this is the minimum number of probes that we would be required to call a change on our array or on our assay. And the next question is, what is being changed? So you want to ask, what is the gene that's involved? What is the region that's involved? Has it been associated with disease or not? And that's how you make a call in terms of the array CGH experiment or assay. So what about resolution? Where does that come into play? So if I were to design this assay, let's say here's one of my disease regions, and here's my backbone, for example. So if I pepper this with probes at every 1,000 bases, for example, along this disease region, whereas the rest of the genome, I'm doing every 10,000 bases. So obviously in the disease region, if my minimum call is based on 5 probes, then probably the resolution is around 4,000 bases, right? 5 probes, you have 4 different intervals, 1,000 bases each. Your resolution in that region is really good, very high. But if I get a call somewhere out here where it's 1, 2, 3, 4, 5, and it's spaced every 10,000 bases, then my effective resolution is 40,000 bases. So it really depends on what you are looking at. The disease regions will be covered very well. I'm not sure if this is the resolution that we'll get on that chip that we are using upstairs, but it just really depends. So it can be better than fish. Even this is already better than fish on a good day. Fish, you may be able to get a signal bright enough at this level to see, but it's very puny. It probably won't be a very good, technically a very good assay to perform, because that's a very small dot to visualize by eye. But this is already better than fish, and this is even way better than fish. This is probably at least, this is already 1 order of magnitude better than fish. This is probably 2 orders of magnitude better than fish. So you get down to very small things. So I would say here, this would be tens of kilobases in terms of resolution. So the advantage of array CGH compared to FISH is that now you get back to the whole genome coverage, right, genome-wide, as best as you can design your probes. So if you have 1 million spots on the array, or if you have 2 million spots on the array, you can make it very, very high resolution if you wanted to. But that's a very expensive chip. So we sacrifice resolution so that we can accommodate more patients, make it a relatively affordable assay per patient on the $300, $400 level. And therefore, we chose this 4x180K chip. But you sacrifice some resolution, but really, a lot of the diseases that we're detecting, which is autism, intellectual disability, congenital anomalies, these are pediatric cases that present with things that have basically gone through karyotype, gone through FISH. Because karyotyping, the resolution is very poor, may not be able to pick up, for example, a 2 megabase region that's affecting the patient. So you get a false negative there. FISH, you have to know what you're looking for, so you can't FISH for 500 different entities. That's just way too expensive to do. So they do something that they think the phenotype fits, but it comes out to be negative. And then you go back and do the array. Now you're doing sort of non-bias throughout the genome, and you will be able to pick up something that's 500,000 bases, or even easily 2 megabases will be easy to detect on this assay. So that's the power of array CGH. Is it gets you sort of the specificity that you get with FISH, but also the genome-wide coverage that you get on karyotype, but also at a much better resolution than you get with karyotype. But there's one drawback about arrays, though. You know what that is?
Mutation.
What's that? You said point mutation? Yeah, well, point mutation you cannot get by nature of the assay. You won't be able to get it by array CGH. But there's another important, so here's patient, here's control. You're going to mix them, right? There's one thing that you cannot detect by array. That you can detect by FISH by design, and you can detect by karyotype. Right, translocations. So you have a balanced translocation. Let's say here's one chromosome, here's another, right? Here's another chromosome, and here's another. Technically, I should draw 4 chromosomes, right? And so hopefully this will be apparent. Right? 2 situations. Patient has a balanced translocation, a rearrangement that's interchromosomal. Nothing happens at this breakpoint. It's a clean cut switching of the 2 arms. You think you cubited 1 microgram of this DNA. You cubited 1 microgram of this DNA. You go do the labeling. Yes, at the molecular level, maybe that junction is covered a little bit differently by 100 bases of that one fragment of DNA that is produced during the labeling, but you're not going to be able to distinguish that on array. So when you go to mix the 2 samples together and you hybridize, there's really no dosage differences between the 2. So array CGH by design is only good for copy number imbalances. But if you have a copy neutral translocation, copy neutral inversion, then you will not be able to detect this by array CGH. It's just not designed to do that. You can hint at maybe a translocation. So typically, a patient who inherits one derivative from a parent, for example. Let's say the parent has a balanced translocation, right? The child's going to get one of these 2. So if you see on array CGH that there's a telomeric loss of a certain chromosome, you may be able to guess that perhaps, yes, there is a translocation involved, but you cannot prove it. Similarly, let's say the patient does have a translocation, and during the process, there is something that's deleted, for example, in one of these chromosomes. So on array, you'll see a deletion. It's an artifact of the translocation, but it doesn't prove that there's a translocation. So typically, if you suspect a translocation based on array CGH results, you need to go back and look at fish, for example, and design it so that you can detect that around that breakpoint whether or not there's a translocation going on. Alternatively, maybe it was on karyotyping, but again, due to the poor resolution, you may or may not be able to pick it up by karyotyping. So for any copy neutral kind of changes, you will not be able to detect it on array CGH. So another question, polyploidy, can you detect that by array CGH?
You're putting in the same amount.
Right.
So control 46, say female, right? And you have here 69 x, something like that. You normalized it during qubit readings or nanodrop. You add, a microgram of this, a microgram of that. Technically, the dosage between these two are different, right? But when you go to do the labeling and everything, and you do the copy number assessment, you basically normalize the two conditions. It's sort of a copy neutral across the genome kind of change, so you will not be able to pick this up. You will by fish, because you're looking in situ at one cell at a time, and you will by karyotype, because technically it's also an in situ assay. Questions so far? No? So there is an advantage of doing SNP detection that I haven't gone into. So there are certain things where during reproduction, during fertilization, for example, things could happen where an allele from mom or allele from dad is lost. So let's just say you have the egg and you have the sperm, right? And they come together to fertilize. The sperm comes to the egg to fertilize it. And during this process, you have some allele. So this is from dad, this is from mom. And for some reason, either this entire chromosome gets lost, or maybe a portion of it, some kind of recombination, some rearrangement or something like that, that deletes this. And what can happen is that there could be a fixing, a repair of this one portion of the DNA. So maybe this one portion, so let me draw this. Say this chunk is deleted. And what's going to result is maybe during the process, this dad's allele is used as a template to fix the mom's allele, some kind of rescue. Like that. So if you were to do array CGH, would you pick this up? No, you won't. So it looks like you still have one copy two, right? At this breakpoint, yes, there's a difference. But when you do the labeling, it has no difference, no regard. Because you're picking up a 60 base oligo or something during the probing, you won't be able to detect this. You still think that there's two copies there. So this is a copy neutral. And what this is called is uniparental disomy. So there are two flavors of this actually. You can have uniparental disomy, and I'm just drawing two alleles here, right? So you can have what is called isodisomy, and this is what is being represented here, is that the same section in the dad's allele is copied over to the mom's allele. So that's called isodisomy. You can also have heterodisomy. So heterodisomy is a little bit complex, but what can happen is that for some reason, this won't happen during the fertilization stage, it might have happened upstream of that, is that perhaps you acquired, I probably need to draw this like this. So dad has, for example, A and a B, okay? Mom has C and D, for two alleles. So in this situation right here, what I've drawn is probably this one portion is A and A from dad. Okay, that's isodisomy. But you could have another situation where you're inheriting this portion from dad, but it's technically it's A and a B. You see that? Both of these alleles are inherited from dad, and that's called heterodisomy. but it's uniparental, meaning it came from one parent. So it's a bit rare condition. Probably clinically we're not even detecting it very well, but at least this portion right here is actually quite important for disease. Because there are certain situations, for example, in imprinting, where if you inherit the same material from one parent versus another, it could lead to disease, where due to methylation effects or differences or dosages, it can lead to disease. For example, Angelman's and Prader-Willi is an example there. Another situation is where you have consanguineous sort of relationships, where the child is a product of two parents who are very related. So what you can see is that there's a lot of regions of the genome that are very shared between the two. It's not due to uniparental disomy per se, but it's because the two people are so related is that there are chunks of DNA in the genome, in the child, that are exactly the same between the two. And what that does is that it unmasks things that are recessive. Recessive conditions where normally if you have one parent who has it and the other who's normal, the normal parent will compensate for the recessive allele from the other parent. But if you have two parents who have both alleles because they are related to each other, chances are 1 in 4, right, that those two alleles will come together and therefore unmask a recessive condition of disease. So uniparental disomy can be detected, but it cannot be detected by a typical copy number sort of array. So how do you detect it? And that is what the Snip array allows you to do. So if you were to design an array that can give you the copy number change, that's fine, right? But in addition to that, built into a functionality into the array that can allow you to do SNP detection, then you can do uniparental disomy at the same time. So the way you do this by Agilent's method is you take the DNA and you shear it with enzymes. So I mentioned ALU1, RSA1. So what Agilent has done cleverly is they selected ALU1 and RSA1 sites that have a SNP occurring there. I forget what, maybe this is what ALU1 is. And perhaps in the reference sequence it's a TAGC, but in the patient we might have a TTGC. So it will cut here, it will not cut here. Right? Because it's like a mutation of the ALU1 site. So cleverly they've gone through and picked 60,000 of these sites in the genome. And they picked things that are in Hardy-Weinberg equilibrium, meaning that perhaps you have some patients, okay, I'm going to draw all the scenarios here, right? Some patients who have TAGC, like that. So if you think about it, this could be an allele state that's A and a B, A, and then B, B. So in Hardy-Weinberg equilibrium, you would expect the two heterozygous states to occur at 25% allele frequency, or prevalence, sorry, in terms of genotype. And you would expect this genotype to occur at the 50% level. So just go back, look in your genetics book, and you'll see it. The reason why they pick Hardy-Weinberg equilibrium SNPs is that this will give you the most informative information in terms of IDing folks. You don't want to pick SNPs that are, very show minor allele frequency of like 1%, because that's not going to be useful for most of the population. Or depending if you're on Caucasian or Asian or African American or African, for example, they will not be informative. So you want to pick things that are as polymorphic as possible in the population, and Hardy-Weinberg equilibrium gives you that. So that's how they select the SNPs. But here you can see, for example, in this state right here, both of these alleles will get cut, right, in the patient. And only one of the alleles here will get cut. No alleles here will get cut. So what happens is that your control sample. Here's the patient, here's the control. And the sample that we need to use is some HapMap sample. that we know the genotype already of. So we know what that patient is at this one locus. We know that they are either AA or BB or AB. We know that for every 60,000 of those SNPs that we designed this assay for. Then you go to do the patient, which is an unknown, right? So you cut that patient with ALU1 and RSA1 in terms of their DNA. You do the labeling. And based on cutting or no cutting, the labeling will be different. And it will hybridize differently to the chip and give you a signature that's either similar to the HapMap patient, or HapMap control, or very different from the HapMap control. And in the end, based on if it's an AA state, AB state, or BB state, you will be able to do something that's called clustering between patient versus the control. And you'll be able to tell whether or not the patient has AA. If the patient matches perfectly with control, then it's maybe AA. Patient kind of matches halfway, which may be AB. Patient doesn't match at all, then so the patient probably has some BB. So that's how you do the SNP genotyping. And the reason why I say it's clever is because already the assay that Agilent performs already requires this digestion step. And they didn't change anything about hybridization, didn't change anything about labeling. I guess hybridization did change because they designed probes to sit right here. That will give you the differential kind of binding and hybridization. But otherwise, everything, the core principle of the assay is the same. The difference with Agilent versus Affy, or Affymetrix, is Affymetrix deliberately designs a probe that's 20-fimer that will recognize this A or this T. So they'll design a probe that has a T here versus another one somewhere else on the chip that has an A, and they'll be able to tell you whether or not it's an A or a B. So it's not based on differential cutting, it's not based on differential labeling, it's not based on differential hybrid. Well, it is based on differential hybridization. So those are two different strategies. But at the end of the day, what you're looking for is a SNP profile for the patient. So here's the patient SNP profile. So you have three states, right? You either have AA, AB, BB. Now let's just say this is 0 cutting, 1 cutting, 2 cutting of the alleles. You could also have a 3-state actually, and a 4-state. So this occurs when you have a non-diploid situation. Like if you have copy gain, copy loss, like in cancer, for example, aneuploidy, you can have this state right here. But let's just look at a germline condition where it's diploid. So typically, because of the Hardy-Weinberg equilibrium status of those SNPs, the chances are is that it's very rare to see someone who most people would look like this, where it's sort of almost randomly distributed. Sometimes it's AA, sometimes it's AB, BB, etc., etc. It's very rare to have a situation where someone would show this. See what's going on? There's a loss of heterozygosity. And why is that? So if I go back and draw you those two alleles, remember this portion is exactly the same from Dad. So probably most of these here, sometimes it shows an A, sometimes it shows a B, sometimes there's a B, B here, sometimes there's an A, there's a B, there's a B, there's an A, there's a B, there's a B here. So it looks like this situation. But within this particular site here, I'm going to zoom in, you probably have a situation where and the reasoning why is that this is the exact same allele from one parent. So it's a loss of heterozygosity or a long stretch of homozygosity. And you detect it by just detecting 0 cuts and 2 cuts, but nothing with 1 cut. And when is this significant? It depends. So there's really no hard cut standard rules for this. But typically if you are at the telomeric end, people say if it's 15 million bases or 20, that's when it's abnormal. And if it's more internal, maybe 10 million bases or so is when that's abnormal. So you can probably sometimes see a short stretch, maybe 3 million, 4 million bases, that looks like a loss of heterozygosity or UPD. But it also depends on the resolution, right? Because we only have 60,000 SNP probes. The resolution is not very high to pick this up. So by chance, you could have a region where, if that region is not very well peppered with SNP probes and you have 10 things that look like this, maybe it's just because you don't have enough sampling. So chances are, yes, you may be able to get a situation that's in a normal case. But absolutely, when you see like hundreds and hundreds of these giving you a 20 megabase region that looks like it's loss of heterozygosity, that's when it's going to look like UPD and will probably be abnormal. For a consanguineous case where you have cousin-cousin, brother-sister marriage or whatever going on, you will see this throughout the genome. It's just 20 million bases here, 40 million bases there, 10 million bases here, just peppered throughout the genome. And that's represented by the crossing over events during gamete formation. But you'll see it just scattered across, and it's very easy to recognize a consanguineous case. So we've run our assay. So far we are just completing, I think, the validation for the UPD, but we've picked up a couple of cases where there were cousin-cousin marriages that show loss of heterozygosity. So this, in combination with aCGH copy number detection, gives you a rather powerful technique. Copy number, I think if you pick the right population in terms of patients, let's say folks showing autism, developmental delay, intellectual disability, congenital malformations, dysmorphic features, if you pick that group correctly, our hit rate so far has probably been around the 10 to 15% range. So whereas these folks probably, karyotype will only pick up maybe 1% of those, 2% of those at most. FISH, depending on what you're looking for, maybe 1 to 2%. Array has actually pretty high yield. So some folks and societies and groups have suggested perhaps that we shouldn't wait for this to be the third tier test, but maybe it should be upfront. The argument in the past was because it's harder to interpret, you pick up uncertain calls. But I think the power in terms of the sensitivity of this assay for this particular type of group, I think at the 10-15% level is very well worth it. It's also an expensive assay to do, but sometimes it really doesn't, a lot of times the pickup that you get is not, it's diagnostic for a condition. Most of the time it's not going to give you an answer about how to treat the condition, because a lot of these conditions affect 5 million bases, 10 million bases. You're not going to be able to repair that very easily. But what it does give the patients and the family is a peace of mind that there is a cause for what they have, for one, because sometimes parents feel like they're guilty, they cause it for some reason. It's merely genetics and the way we are and the way we evolve, and that's how it explains the disease. But it gives the family peace of mind that it's not an unknown entity. Secondly, what you could do is direct patients and their family to support groups that have similar kind of scenarios, so they can basically get information about what to anticipate in the development of the child. Maybe at age 10 you might want to do this, you might want to consider this service or that service. So it helps manage the patients that way, but it won't cure the disease. And then finally, for the family, if they plan to have a second child, for example, or a third child, by using this information and testing the parents, you might be able to say whether or not this change will recur at a certain percentage or not. So if mom or dad has this sort of change, and it's what is explaining the disease in the patient, then you might have a 1 in 2 chances of having another child with it. But in other situations, if it's de novo, where you only find it in the proband but not in the parents, then it may be almost a 0% chance that this will recur. So that's for copy number. So for the UPD component, I think it adds another 1 to 2%, because those things are quite rare compared to copy number change. So it does give some added yield, but it's not going to give you 5 or 10% more. But we typically use this, again, like I mentioned, for pediatric cases, developmental delay, autism, intellectual disability, and multiple congenital malformations or deformities. Yes, questions?
I know that some people are considering using that array also for cancer.
Right. So cancer, obviously, it would be very crazy. So if you've seen any arrays on cancer, you'll get aneuploidy across the genome. Sometimes 10 chromosomes are involved, whole P arms, whole Q arms. smaller deletions, EGFR amplification, easily detected by array. So array, I forgot to mention that there is some sensitivity issues with that. So you need to have roughly, my guess from what I hear, is about 25 to 30% of disease cells in order, minimum, before you can detect it. So in this case, I'm sorry, this is allele at the allelic level. So you need to have 20 to 30% of the allelic change in order for you to detect this. So if it's sort of a heterozygous call or a 1 in 2 hit in a tumor, you're looking at 50 to 60% tumor cellularity that's required. So yes, you can use this to detect it if you have the sufficient amount of tumor going into the assay. But the challenge for cancer is that you get so many changes that you don't really know what to say about it. So if you get 10 different changes, whole chromosomes, whole P arms, whole Q arms, whole genes deleted or duplicated or amplified, you really don't know what to say about it. So right now the challenge is how do you interpret that data? For pediatric cases, usually it's just one change, maybe two, and of the two, maybe one is abnormal, the other one is uncertain, and is a much weaker call. It's a little bit easier to interpret that compared to, say, for cancer. So for right now, I'd be a little bit anxious to launch it for cancer unless we had maybe a legit database or more information about what to say. For certain diseases like multiple myeloma where things are predictable, yes, you can say certain things about them where it's known. And it's very finite in terms of the number of calls. But otherwise, no, I wouldn't launch it for solid tumors quite yet. So I think we should stop.